In this session, we will learn how to compare protein sequences from a variety of species using Megalign. In order to do this exercise, it'll be useful to have the human protein sequence for your gene ready, and also to download protein sequences for your gene from at least 4 mammalian species. Here are a few examples that you might wish to try: the rat, the mouse, chimpanzee, pig, or cow. Let's take a look at how you might download the protein sequence for the chimpanzee. If you go to NCBI and go to the protein section, and we'll use our example CFTR again, type in CFTR. But you see that that gives a huge number of results. So let's narrow it down a little bit, and we'll pick gene symbol CFTR, or gene name. And then for this field, we'll put organism. And we know that the chimpanzee is pon troglodytes, so we'll just put in pon, and that'll bring in a much smaller set of results, the first of which is the chimpanzee. So we'll download that file by choosing the send to option, and we'll send it to the FASTA format. So we'll click file and then change that to FASTA, and then we'll download that and use that in the alignment. So I've downloaded the protein sequences from a number of species and put them into a folder. I'm simply going to select all of them, drag them into the pane of Megalign, and then pick protein as a project type. Then I'll go to the align menu option, and I'll pick the first option, Clustol Omega, and that will align the protein sequences from these species. Once the alignment's complete, one thing I notice is that the file names are the NCBI ID numbers, but I want to name them by the species. So each time you click one, you can see the species name, and you can go through them. So that was the pig, and then you can just go through each of them and rename them. So I'm going to go through that really quickly so that my alignment results will make more sense. Instead of using these long ID numbers, I'll see the species name that I'm working with, and I'm using the common names. So once I complete that, then it'll be much easier to work with the files. And you can see in the bottom right, it will, for each file you select, it displays the species name. So the last one is the mouse, so we'll go ahead and change that to mouse. And then for good measure, I might also want to change the name for human to show human. And one thing I noticed is that the rat sequence has a long beginning area, and that's probably going to throw off the numbering. And let's look for our favorite position in CFTR 508, and we can see that it's not at position 508. But in other exercises, we learned how to recognize that using the KENII preceding amino acid sequence, and it found the phenylalanine here. We can see that it is conserved among all of the species evaluated. Now we're going to clean up our display a little bit, and we'll change it so that we'll only color the amino acids in any given species that differ from the consensus sequence. So we'll make some changes, as you see here, and that will change the display so that now only the positions that differ from the reference sequence or from the consensus sequence are highlighted in the background. You have a variety of other options, and you can experiment with these presentation selections as you see fit. So now let's change the view, and you can see there's a variety of options to work with. To change it to a tree view, which is what we'll do, we actually go to the bottom and pick that option, and it shows an evolutionary tree. As you would expect, the human sequence is most related to the chimpanzee. The baboon is close. The mouse and rat are closely related to one another. The pig is definitely distinct from the rodents, as you can see through this tree. So we've seen how to load a variety of animal sequences into Megalign. This table just shows numeric representations of the evolutionary distance between each sequence. So in this session, we've learned how to use the DNASTAR application Megalign to compare protein sequences from a variety of species.